The preclinical phase would take anywhere around 5 to 6 years.
That is when the AI bug bit us.
So what got you into drug discovery kind of
So the traditional drug discovery, the early 2000s, there was a tool called computational chemistry that was being done. Technology has moved from just looking at traditional drug discovery and doing rational drug design to a more computer-aided drug design to now a more AI-aided.
You understand the protein model, you understand the protein.
A disease, it's a chemical reaction gone wrong. And it's an over-engineered system.
Whatever we did maybe 15, 20 years ago has resulted in something that is actually saving lives today.
Finding the right molecule to modulate it, I would say that is engineering.
Welcome everyone to this episode of the Double Down podcast. I am here at C Camp in Bangalore, which is the Center for Cellular and Molecular Platforms. And I'm here to actually play a game with biology. I'm going to be discussing a very interesting project on AI drug discovery with a company called Peptris. And I'm amazingly lucky to be joined by Venkat, Anand, and Sridhar, the founders of Peptris. And we'll talk about how AI is going to play Tetris with proteins. So with that, let me welcome you all again to this Double Down podcast, and thank you so much for doing this on a short notice.
Thank you, Chinmay.
You know, it's really been fascinating to see how some interesting frontiers are being handled in India, specifically in areas that India has not been known for in the past. And for me, what was very exciting was one, the drug discovery part is probably well understood. It's been there for many, many decades in the world. But it's the whole concept of multidisciplinary approach that you bring in to this game. So, you know, what I'd love to do is to kick it off by actually getting a little bit of a background of each one of you, and then we will start to kind of delve into the details of what Peptris is all about. So, Venkat, maybe I can start with you.
Excellent. Hey, thanks Chinmay for doing this. I'm Venkat. I'm the CEO for Peptris Technologies. And of course, for my background, I passed out of IIT Delhi in '96 with a degree in math and computing, and was working happily in the software industry for quite some time until the drug discovery bug bit me. Yeah.
Okay.
And that was about 7-8 years back now. And from that, there has been no looking back. It has been an amazing journey of learning every day, learning from amazing people around. Trying to understand what is actually happening inside the human body. That has been an amazing journey, and of course, I would love to. I think you should hear about the other co-founders here who are with me.
Thanks, Chinmay, for calling us.
Thanks,
and, my name is Anand. I'm the CTO for Peptris. As Venkat said, I'm also an engineer coming into this new game as you described. It's very fascinating because a lot of technological advancement we see throughout the world, but there's a cross section which, when it finally meets, it gives a very big leap to all of us as a humankind. And we're very happy to take this forward in our own means.
No, great. So you are the two engineers on this side of the table, and now we move to the biologist, the purist.
Thanks for having us here, Chinmay. So my name is Sridhar Narayanan. I'm a pharmacist by training. I did my B.Pharm from Mumbai and then did my PhD in pharmacology from Ohio State University.
Right.
Did a postdoc from UCLA in neuropsychiatry before I came back to India almost 25 years ago now, and have been part of drug discovery journeys with various organizations, starting with Piramal's, Glenmark, Dr. Reddy's, Orchid, and my last assignment was head of R&D at AstraZeneca, leading their drug discovery efforts in the neglected disease space.
Right.
When they closed down in 2014, I started my own foundation called the Foundation for Neglected Disease Research, which is an organization which works on infectious diseases which have a high social impact, diseases like tuberculosis, diseases like rabies, which not many companies in the world want to work on. Till the time, I met Venkat through our children. Both Venkat's son and my son go to the same school, same class. And,
Very interesting.
We used to meet frequently outside birthday parties, and conversations began to move from just family to, the profession, and discussing how They were using AI to do a lot of work in the large screen display space. Uh, I myself have been very interested in how technology kind of intersects with biology, and I'd been reading about AI and its impact on drug discovery, which was just getting started in somewhere around 2015,
Right.
I think, and some of those meetings started growing into much more foundational discussions on how AI could help with drug discovery,
Sure.
and how some of the work that they were doing with their careers
Right.
could potentially interact with what I was doing in my career.
Awesome.
Right.
Awesome.
And that's one thing led to the other. And I think over a period of time, Peptris was established.
Oh, wonderful, wonderful. In fact, I'll go back to that theme a little later down in the podcast. But I think coming back to the origin story, I think, just want to kind of lay the framework here. So how did it actually happen? Like that movement to drug discovery from what you were doing, and then I think you were doing a lot of work on imaging, right? If I'm not wrong.
Yes.
How did that world collide? Like how did this team come together, and then how did these missions kind of align? Two engineers and then one biologist and then other folks and a data scientist who's not here, but we know about him, but came together.
So interestingly, so it has been like 25 years that my association with Anand is. So it's not like it was we met for this mission.
Right.
So so I was at Philips Software, basically trying to build televisions. Yeah, and that is when. That was the age when if you I told somebody that a television has software inside, people never used to believe it.
Right.
Everything looked like a mechanical device. Software was like the Y2K problems are probably the only things that were known.
Right.
Or building large systems and so on. So when you say embedded systems and so on, it is still not known. So we were working on these devices, and Anand and Amit, the other two co-founders, joined my team at Philips. And we were working on televisions, basically software inside televisions, and we saw that.
So basically, the journey to start and convert smart TVs and have smart TVs happen.
So basically, it was going from the analog television to the digital television.
Right.
After the digital television came your smart television.
That's right.
So we saw that journey as in like. So now if you talk about software in a television, everybody understands. So you need to download a Netflix app to watch something.
Yeah, or tell Alexa to load this or whatever.
Alexa
Right.
to load this, everything is fine.
Correct.
But even otherwise, like right from the first remote controls, the way they work to what it is. So we saw that journey. So in real sense, what we saw at Philips, like right from say 96 to 2007 when I was there, and probably 7 years that Anand and Amit were there, we saw that transition happening, how technology was actually reaching out almost to all the kind of devices that we were looking at and our interactions with the world. As in like, we were looking at better interfaces, and that is when the next big moment happened. Of course, the mobile phones and so on. And we were building, and we moved away from Philips. Philips was, I would say, a place where we all learned. So at least when I say we all learned, it's me, Amit and Anand, where we learned to build systems to scale. Basically, because any software that we were running was going to be deployed on millions of televisions.
That's right.
Yeah. So we knew what it means to have a fail-proof software. Nobody talks about a software crash in a television.
That's right.
Yeah. So building robust.
Talk about a blue screen on a television.
Nobody talks
blue
about
screen
it.
of death.
Correct. So in that sense, building systems that are robust, building systems that run, run. You don't need to think about it. For the next 10 years, it should run.
Right.
Yeah. So we had done that, and that grounding, I would say, comes from our Philips days. And from that, we moved to a Valley-based startup, and that is where I would say our next phase of our professional career started, where we started about dreaming big. Yeah. So in real sense, we were building a large panel display, completely new technology called laser phosphodisplays that Anand I think will talk about later. But it was more about building something from scratch, that we can build all the hardware, the software, the systems to hold it, everything from scratch.
Very nice.
And we did it. That is a Valley-based startup, and a good part of the engineering, I would say a major part of the engineering was done out of Bangalore.
Wow.
Yeah. So it was a small sleepy place called
At that time, we
Rajajinagar.
didn't have a word called GCC to maybe define it. But you were probably at GCC, right?
We were at one level, but we were more like we were building a product from scratch.
The full
we were
R&D
not
and the team.
We were an R&D team come this one. So we were not a services arm to a larger organization and so on. So in that sense, it was a much different setup.
Sure.
And also we were not talking about numbers. At the peak, when we had recruited everyone, we were about 14 of us, including the office boy who was sitting in the front. Yeah, so we were 14 of us. And what we were building was not just the hardware or the software, we were building systems. So we were designing right from the packaging to the display systems, the interconnects, everything.
Wow.
And the good part of it was there was a big team out there in the valley also, which were doing some parts of the tech, and we were doing the some parts of the tech, and we were constantly integrating it and we're making it a larger system. That gave us that what do you call, the brush of this one, saying that yes, we can do something from scratch, we can do something much bigger.
So that's the beginning of your entrepreneurial kind of bug.
More or less. That I would say is the seeds. The seeds were sown there. And it's a company called Prism Displays.
Okay.
And the 5 years that we spent there, that was a transition for us, as in like from being in a GCC, like Philips in that sense was a GCC,
right?
Like to doing something
in a more startup environment,
building from scratch and so on. So there you didn't have any borders. You couldn't say that that team didn't deliver, this team delivered. We were the team that was delivering.
Everything was on us.
Everything was on us. Yeah. So
perfect.
that way and probably that carries off, as in like in terms of, because we, at Philips also I would say it was a very good part of it, but it was only the software part of it that we were dealing with.
Got it.
This was a complete systems one. And if you want to talk about the display itself, that will be good.
Yeah.
Other than what Venkat talked, right?
Although we want to focus on AI for drug discovery later, but absolutely.
But the idea, at least in the Prism displays was based on completely a very new technology called laser phosphor display. We had cathode ray tubes where we used electrons to throw an image on the screen.
Yeah.
Instead of electrons, we used a laser. At that point, controlling the blue ray, which is a blue laser, to create an uniform image was hard, and it's a very, very new technology. And as Venkat said, the experience at Prism gave us that, ideas about when you want to solve a problem, you have to go down to first principles. Yeah. That's when you really understand what is actually happening. And luckily, the team there in US were very sharp, helped us to a great extent. But at the end of the day, we did deliver something, and that's what we are all proud of. And that journey is what still tickles in us, and we continue doing whatever we do.
Got it. And so, what got you into drug discovery kind of story? So that's the interesting part.
Yeah, that's interesting. So after that, so all this is about like having to use building systems, building larger systems, using data to analyze stuff and so on. The image processing, basically getting uniformity and so on. So all of it was happening.
Right.
And we slowly moved to what we call as device diagnostics using data.
Yeah, and Philips is a big brand for that as well.
So we went back to Philips and we were doing work for Philips again as consultants. And one of the key programs that we were working on is how to do diagnostics with all the data that is generated, along with, of course, building the first Android televisions and so on. So at that point of it, we really found that some of the things that we had been doing for ages, suddenly AI was coming into those fields and actually making us rethink as to how do we really solve this problem. There seemed to be a much better way to solve this problem. That the first ImageNet software came out in 2012, and it kind of revolutionized the field that we were working in. For example, at some point of time we had thought about how do you find whether a logo is on a television screen. Where is the logo? If I had to do it the hard image processing way, it took us a lot of time, lot of effort, and a lot of characterization before we could say that, look, the logo is in this part of this input signal that is coming in. Plain, simple logic. I'm not talking about hardcore science here. As a human, you see it and you know where the logo of that TV channel is.
Correct.
Yeah?
But for a software to know
but
where
for
it
a
is.
software to know it and with plain image processing, because you're going to see bits and bytes of it.
Correct.
How do you do it? And then walks in ImageNet. It's talking about, is there a cat in the picture? And it will tell you. You're saying, pause, we're not able to find a logo in the picture so accurately. And here comes an algorithm down the path and it's able to differentiate between a cat and a dog. There is something fundamentally wrong in what we are doing. Yeah, and we need to rethink about the problem completely. And that is when the AI bug bit us.
Got it.
And one led to the other. And we started reading more about it, we started dabbling with it, and of course, it was a natural progression for us because the same problem that we were solving were being done by the first wave of algorithms, much better way.
Right.
Yeah.
Right.
And I think I wouldn't say that one day we got up and we say we will start working on drug discovery.
Right.
That is not it. That's definitely not the way it happened.
Right.
Yeah? And of course, as I said, 2011, 12, when this realization was happening, saying that there is something big happening in the world, yeah, saying that a lot of things are changing. At a much more rapid pace than we had seen. So we had seen the first 15 years of the digital transition.
Right.
But what we were seeing in 2012-13 was a lot different than what it was.
Understood. Yeah? There was a step change.
There was a step change.
Yeah.
And one of the things is that, of course, a lot of compute was required, and that was not what we were doing in the embedded world and so on. But of course, it was all getting available at the same time.
Right.
And meetings with Sridhar and a lot of discussions with him during the 2015-16 time frame.
Because he was probably solving that problem from the other end, right?
He was talking to us. I used to ask him, what is big in drug discovery? So when you say, if you have to look at it like, look, I am able to say that like there is AI happening. He was telling me, you should go and read about CRISPR. You should see what it is, how it is revolutionizing.
So maybe we should pause there and talk to Sridhar a little bit about this whole traditional drug discovery process, because that world, I think, is very important from where you're coming from.
Correct.
Right. So maybe a few inputs on what that world looks like.
Yeah, so if you were to go back and look at what traditional discovery or how companies performed traditional discovery. So if you look at the multinationals, right? Multinationals would have libraries of compounds, millions of compounds. Right?
These are like pre-synthesized molecules.
These are pre-synthesized molecules. So they would make molecules for projects, and each of those molecules would get tested whenever they would have a new target or a new disease that they wanted to tackle using a specific protein. So they would do this something called high throughput screening. So they would develop a biological assay which would analyze for that particular target.
Right.
And then they would screen a library of compounds using robotic systems to kind of understand whether a particular molecule works on or modulates that particular protein target.
Got it.
And this was time consuming, it was cost intensive, and they needed to physically have the library to do this, right? Now they would find out of a screen of 1 million compounds, the hit rate was somewhere around, I would say, the best case scenario would be about a 1% hit rate, right? So you would get 100, and when I call something a hit, it is at 10 micromolar kind of concentrations. It's able to modulate the protein, right? And that hit rate would be the starting point for new chemistry to evolve, right? So they would take that particular hit molecule, work around it, create new IP space, and then work on that particular molecule to move it through the development chain, which itself was time consuming. So the traditional drug discovery, the preclinical phase would take anywhere around 5 to 6 years, right? And the whole clinical development would take another 5 years.
5 years.
So you were looking at, from a project start, about 10 years to get a drug approved on the market, right? And so that was a lengthy, long, time consuming, costly affair.
Right.
Now, once AI came into the picture, the thought process was this would really shrink each aspect of
which
this.
Was technically when? Which is the CRISPR movement, or was
Not
it like
the
a
CRISPR
movement before that?
I think 2014, 2015 were when one of the first, the serious papers of use of AI in drug discovery started coming.
Right.
So there were, I would say, even in the 2000s, early 2000s, there was a tool called computational chemistry that was being done, or and people would look at protein structures and then look at a binding of a molecule to that protein,
right,
using different software. So Schrodinger was one of the main leaders in this, but that didn't give a lot of success. There were some successes, but it was not as successful as the technology was touted to be, right? Now, when AI came in, it started combining a variety of these abilities in terms of software,
right,
to kind of come out with faster methodologies, more efficient methodologies. And today, I would not be surprised with AI companies talking about hit rates of about 10% is a given,
right?
So I think that technology has moved from just looking at traditional drug discovery and doing rational drug design to a more computer-aided drug design to now a more AI-aided.
Fair enough. I think that's a good way to frame it. But I think one of the other things that are kind of still stark in this whole picture is, when I talk about we're sitting in 2026, we were discussing this, that so much of technology is already out there. But when it comes to the human body, we still understand very little.
Correct.
And I think there is this very interesting thing that, you have this concept of human proteomes, right?
Yes.
Maybe if you can just throw some light on this, because I think this is very important to understand literally what percentage of druggable candidates practically are out there which are not yet discovered. That universe is huge, even though we have done so much of work already in different areas. So maybe if you can just explain for us, what does that human proteome model looks like? And then where are we sitting in that chain?
So let me go back a little bit.
Yeah.
Right? So most people.
Because this is the drug discovery opportunity.
Correct. So most people are very familiar with the Human Genome Project.
Right.
Right.
That's
and,
right.
It was slated to be revolutionizing how healthcare was going to be delivered.
That's right.
Right. And once we had the entire human genome decoded,
right,
everybody thought that that's it. Now we are going to have
the key.
We have the key. We're going to treat every single disease on earth.
That's right.
Right. And 25 years down the road, we are still not able to do what we wanted to do with the Human Genome Project, right? Or use that data more efficiently.
Right.
Today, we have got technologies where we understand the proteins in the body very well. We understand the metabolites that are formed, what are the endogenous ligands. There is a significant increase in knowledge in the last 2 decades, right? However, we are still only scratching the surface, right? Because genetically, there are significantly different pools around the world, different diseases around the world. And our ability to understand which proteins are responsible for what aspect of the disease is still at a very early stage, I would say. While we understand certain diseases better than others, majority of the diseases, our understanding is still quite minimal, right? And which is why, though we understand what proteins are involved, the causality of those proteins or the directly linking them to the disease itself is still a journey which takes 8 to 10 years. You have academicians. There are some academicians who would work on a particular disease for life and still not be able to, understand it in its entirety.
Right. So these are going to be like, and just correct me if I'm wrong. So these are like, you understand the protein model, you understand the protein, and then you see how this is, any change in that protein structure, what kind of symptoms it's leading to, what kind of diseases it's manifesting in, and then what kind of outcomes it's leading to.
I'll give a very different perspective on this. Okay?
Yeah.
So what happens is, like, there are, it is like you mapped a complete car, say, all the parts of it, but you still need to know how it works.
The car, right?
The car.
Yeah.
You can say that I have mapped all the nuts and bolts of it. I have mapped every.
I know everything.
Everything about it.
Yeah.
But you only know the nuts and bolts. You still have to figure out how it works.
Correct.
Yeah. And what is really happening, and typically that is what is happening with the human body also. Like, we are a very complex chemical reaction at the end of it. The way we think, the way we speak, the way we hear, everything is a chemical reaction finally.
Right.
When you think about it, we are just a bunch of sensors wired up together at one level, and each of these sensors work in a particular way, in a biochemical way.
Right.
In that sense.
Right.
And when you say a disease, it's a chemical reaction gone wrong. Yeah, or it's something is being overactive, or something is being suppressed, or something is not at all present, or things like that. And how do you correct it? So technically, like what we used to say, that look, at the end of the day, the biologists are like master storytellers at one level. Yeah? So it is like seeing the poster of a movie and telling you the whole story.
Right.
Yeah. So what we have done is we have mapped, as in over time, done all these individual small pieces of things that are working down in a car, and we think that
so
we know
the
car
part.
runs.
We know the part.
We know the parts. But unfortunately, unlike things that humans have built, what we are is a very complex system, and it is an over-engineered system. Yeah?
Over-engineered, okay. God or anybody else, please correct us whether over-engineered.
Well, over-engineered because most of the time when you talk about there is a disease, so it is like after a lot of things have gone wrong, because there are a lot of mechanisms inside which will try and fight to get back the normalcy. There are a lot of competing systems inside,
Right.
which are trying to get back the rhythm, get back into that same harmony.
Yeah.
And we are kind of like trying to understand pieces of it, and we probably understand one pathway better, and we say that okay, this is what something is happening, and we go and try to change it.
Right.
And then another pathway, which is a compensatory one, kicks in and it does something good or bad. And biologists are constantly grappling with it, with new information about all this dynamic behavior that is happening within the system. And that is why when you say, when an engineer says that okay, all diseases will be solved in the next 10 years, the biologist will say, oh, we don't understand anything, so wait.
Yeah.
We will solve things that we have a better understanding of. And that is about 10% of the system.
This is such an interesting point, because when I was thinking about this problem a little bit, and then when you know that AlphaFold cracked this particular thing, and I'm sure you have some views on it, and would love to hear what your thoughts are in terms of what it triggered, and then finally ultimately Demis going and winning the Nobel Prize for chemistry, right? It's a different universe, but the problem they're tackling is similar in terms of just the
Getting all the building
interaction,
blocks right.
the interactions that are the systemic complex things that we don't understand.
True.
Right?
So basically, AlphaFold is talking about all the individual nuts and bolts of the system, how they could potentially be
Leveraged and used to maybe
Not even leveraged and used.
leverage, not leverage.
All they're saying is, here is a protein, and this is the kind of shape and structure of the protein, right? Beyond that, I don't think they're telling you what is the interaction.
What's the interaction?
Right? Or what needs to go up, what needs to go down. That is not what AlphaFold is doing. It's telling you more about the protein itself.
So this could be like that complex molecule library that used to exist. If they've given you the library, now you can do something with it to figure out, and there is a whole world that you have to do with AI
again,
and exhibit.
This is only a hypothesized structure, right? It is not the final structure, not the final crystal structure, right? And the funny part about these proteins are they are not in a static form, right? So it's not like a lock and a key, just like a lock and a key, right? The lock is vibrating, the key is vibrating, right? So you're looking at trying to get into a lock in a vibrating system, which is not the easiest of things.
Right.
So our understanding of the protein structure is not a static one; it's a dynamic one, right? And AlphaFolds today, I think, is still at a static level, moving towards the dynamic. But I think over a period of time, we will understand some of these proteins better. We will understand families of these proteins better. To come up with drugs for each of them.
Right. So when you come back to this whole, human proteome project, and then the whole, 20,000 plus proteins, I think only 19,000 of them are probably understood a little bit. And then that's what I was reading. I may be wrong. And then there are about 2,000 that are not probably, we have no clue about. And then in that entire universe and subset, this, explain to me, what does this mean that 90% of druggable targets have no known drugs? What does that really mean in simple terms?
So, in simple terms, you don't have a molecule that can actually modulate that protein. Or you have locks, but you don't have keys which can open them.
So the locks are there?
The locks are there. We understand. Today with AlphaFold, we have said, okay, there are so many locks. But we don't know which key will open it. Right? And that's the uncharted space that is available for us to work on.
Right. And now it makes sense in my head, because now I can think where this massive problem of computational combines with biology to help you discover that key for that lock.
Okay, so now when you...
Sorry, I oversimplified it.
Yeah, no, when you double down on it now and say that, okay, what is the space of keys? As in, how many keys do we have? How many keys do we need to figure out? That number is an astronomical 10 power 63. in that
Yeah,
order.
right.
We may be, there are different, this one it can be 10 power 60 or 63 or
It
whatever.
doesn't matter in
But
the
it
relative
doesn't
scheme of
matter.
things.
After,
Number
you have done a
Yeah.
50 zeros, it doesn't matter whether there are 2 more or 3 more
That's
zeros
right.
there.
That's right.
Okay, so that is the total number of possible keys that we are talking about. We are not talking about keys existing.
Yeah.
Keys that could potentially exist. Yeah, and now you are tasked by saying that, look, there is this potential lock, because we are also not telling this is the lock, this is the exact final shape of the lock and so on. A potential lock with its current shape. Can you find a key from this 10 power 60 number which will kind of modulate, which will unlock this particular lock?
Got it.
So this problem is very massive. So you are not going to put a farm of servers and get a solution for it,
Yeah.
because you just can't even enumerate it.
Right. Right.
Yeah. So
Right.
that is why AI is very much needed, and so are very smart algorithms and smart ways of characterizing the problem better for you to find a solution. Yeah.
Right.
And that is where I think our primary work is. As in, how do you characterize these molecules better? How do you ensure that you can get to a probable key? Nobody is trying to find out the key. We are trying to find out
The probable key.
A good key. As in,
Candidate.
like, it is not the best key. It is not something that solves it. This one. We want to know a candidate key which can potentially do some interaction, some modulation of the protein of interest. To even get to that is a very, very tough challenge.
That's right. And that's where you have chosen to focus on as Peptris.
As Peptris.
That this is the game that we will play. So essentially, you're, to simplify the game of Tetris, which is basically saying you can rotate the blocks to a right sequence to make it land straight
Land
and
on that-
complete the sequence.
complete the
So you're basically saying, okay, I'll give you the molecular
knobs.
candidate that will possibly fit into this sequence and therefore result in a positive outcome or health outcome.
That's why you get more score.
It makes a lot of sense.
To fit
Yeah.
the team correctly, the
Yeah.
molecules are tetrominoes for us.
Hmm.
Tetrominoes are the ones,
The
small blocks you
blocks,
can
correct,
rotate, play around.
correct.
The big pile is a protein
Right.
or a lock, as we call it.
Right.
And our intention is to make sure that molecule fits in.
Right.
And makes it complete for a protein to be modulated.
Oh, very cool, very cool. Now, before I kind of now dive into the whole Peptris story and now get into the specifics, I have one question for you, Sridhar, which is more around the fact that, again, going back into your world, you were the co-inventor of N-metazobactam, you were co-inventor of MMV253 and TBA7371, which is like, and you can simplify to us in what disease areas these focus on. What were your key learnings coming out of that world when you went through that process of actually discovering these
Right.
or inventing these molecules?
So I think, first of all, there are very few people in the world, I would say a few hundred thousands maybe, who have actually contributed to the discovery of a drug.
Right.
And to be part of teams that have discovered some of the drugs
Right.
is a significant achievement in itself.
Right.
And again, drug discovery is a team game. It is not one person saying, either a chemist or a biologist saying, oh no, I have discovered this drug.
Right.
Which is why there is a lot of co-invention that happens.
Right.
So I'm a co-inventor on N-metazobactam, which is now an approved drug, and
Right.
that's the only one.
Right.
Right. The others are still.
And that treats what?
That treats it's for bacterial infections.
Bacterial infections, right.
Right. And the others are still in clinical development. Right. So I don't think that it still qualifies as a drug. So with N-metazobactam, this is from a company when I was at a company called Orchid, right? Orchid Chemicals and Pharmaceuticals based out of Chennai. This molecule was actually synthesized before I joined Orchid. So the quote unquote discovery of the molecule happened before I was
Sure.
I was there.
Sure.
But under my leadership, we actually moved this molecule into the disease areas and into development.
Right.
Right. So while they had tested it for a few of the bacterial infections, I think 2008 was when we kind of went and filed. 2008, 2009 was the method of use patent, which actually ended up being the patent which attracted a partner company to come and work with us.
Got it.
So my team delivered the preclinical package on this, and we moved it all the way to a clinical candidate stage, which means that this was ready to be tested in humans. So it had cleared all the safety, all the toxicities.
So from in vitro to in vivo.
In vitro to in vivo to safety, toxicity, all of it completed and being a clinically ready candidate. Right?
Got it.
That's when a company called Allecra licensed it from Orchid, and they took it further into development, doing the human phase 1, phase 2, phase 3 studies,
Right.
and actually getting it into the market. So while the journey of the molecule started in 2004, it actually became an approved drug in 2025.
Wow.
Right, so it was a
20-year
very long journey for the molecule, right? And it's an achievement because that's the first drug from India to have global approval.
Right.
It's approved in US, it's approved in EU, it's approved in multiple places in the world, including India now.
Right.
Right, and it's being marketed in India. So as a scientist who's worked on this is extremely satisfying to know that whatever we did maybe 15, 20 years ago has resulted in something that is actually saving lives today.
Right.
And there is nothing more gratifying than that.
Right. And in terms of learning, so I would say so the aha moment there for you is you have done this for a 20-year period, you've seen this as a 20-year journey.
Yes.
Now the ability to compress this and win the time on this, because for anybody who's fighting a disease, any time gained is
absolutely vital.
Absolutely, uncomparable to anything. Is that the key learning that you can compress time with all these technologies now, or you can do much more, because now the number of candidates can also be very, very specific, right? So the AI is now generating network effects in drug discovery as a process in itself, right?
Correct. So I would think for every success that people have with a drug, there are probably thousands of failures.
Yes.
And the learning is not only from the successes, but learning is also from the failures as to what went well, what didn't go well.
And that is also data.
That is absolute data, right? And actually, when we build our algorithms, we take into account the successes, we also take into account the number of failures. Right? Because that's how the algorithm learns what is good and what is bad, right? And be able to predict it.
Got it.
So from a perspective of the knowledge itself, right? And I frequently use a term called augmented intelligence, right? It's not artificial intelligence, it's not human intelligence, but humans adding value to what is the output from AI.
Right.
And I think that is where the difference for us as an organization is, right? You build really very good algorithms based on small data. So the data sets available in biology are very, very small. I think Anand can tell you more about it.
Right.
But using that small amount of data to come out with algorithms which are predictive to a reasonable degree is very important.
Very important.
And for us to be able to apply our biology knowledge on top of what the algorithm is telling us is what differentiates us as a company.
Right. And that's why this match made in heaven has happened for Peptris.
Absolutely.
Awesome. Perfect segue to now get into the Peptris model. So, maybe Venkat and, Anand, you can probably talk a little bit about the discovery to license kind of journey and the Peptris model specifically there. Anybody?
Yeah. So I'll take a thing.
Sure.
I think initially when you started discussing with Sridhar, it's very clear that finding a drug is a hard problem, takes a lot of time, and of course has a lot of failures, right? So if you want to make a company to find a drug, you need a lot of good funding. I mean, you can't say, okay, with a very small amount, how can I do? There are so many steps to be done. So one of the key fundamental problem was we need to get away from need of large funds, because if you don't do it, as a company, we may not survive. Right? That's survivability is a very key here.
Yes.
So that has been the driving point for us.
Okay.
Now, if I use AI, I can do a lot of processes, things to reduce the cost and the time, of course, but it still requires a process-oriented thinking to reduce the overall cost. It is just not that an algorithm gives you an edge and then you go and solve the problem.
Right.
So initially, when you really looked at it, we found ourselves in a very engineer way, because we are coming from the semiconductor world, embedded system world. We had seen a fabless model there.
Right.
And we thought, okay, there is a very good case for being a fabless drug discovery company. I put the fabless in quotes. It will be a labless, kind of
Okay.
stuff. And we really wanted to say, okay, if we cut down all my capex,
Yeah.
what is core remaining is the IP part, and let's invest everything there, right? So I don't need a large capex to build labs. We invest everything to find drug, and whatever is required to find that, let's outsource. If you think about that, then there is a way for us to move ahead in drug discovery without the baggage of a lot of labs, a lot of people skilled in conducting lab experiments and all that.
You don't need to have the end-to-end stuff, and
End-to-end
now you
stuff.
can truly collaborate in meaningful ways.
Correct.
Right.
And that money is required really when you reach that stage.
Right.
Among all the failures you will see, the first time, as you said, trying to find that one key that might possibly work is the hardest. You don't need labs there to an extent where I need to put a lab, put people and do everything.
Right.
So we segregated the problem. We said, okay, let's compute do this problem and outsource this part to somebody who can tell us where this becomes a key. Once that is done, the other parts can already be put in place. You put in more money into it, and
Right.
hence we can successively invest to get a drug. And that is how we view ourselves as a fabulous drug discovery company,
Got it.
trying to make a very difference with a very limited amount of money, but the aim is to get a drug so that patient is benefited.
Fair enough. Fair enough. So if we were to just like simplify it from like the traditional model, right? Like you have the disease hypothesis, which basically helps you discover what are we solving for. You take the indications, then you identify the targets. So the Peptris model is you take targets that have been pre-identified, and then you go into early discovery to optimization to preclinical.
Right.
Is that the phase that you focus on, and then you have the rest of the stuff, which is client development, the actual distribution and marketing, commercialization. That is all
Okay,
anyways
so-
outside. So
yeah.
like, how do you split that whole journey? I'm
Okay.
trying to figure out like where does
Where
the
does-
essence of
But
Peptris
if
lie?
you really look at it from an essence perspective,
Yeah.
the way we look at the drug discovery was in, like for example, right from disease hypothesis to drug in the patient's hands, that journey is much longer than a decade, as in like
Correct.
it's about 2 decades.
Yeah, we know that. Yeah.
So some of the targets that we are currently working on, are probably like discovered a decade back, saying that yes, this protein is implicated in a particular indication and so on. So I would say that part is science. Then the next part is, once you have a target that you need to modulate, finding the right molecule to modulate it, I would say that is engineering, because here it is a search and a sort problem. You what do you call, assemble 3 guys from the computer science world and put them in a room, they'll say everything in the world is a search and a sort problem. And we look at the world like that. Okay, so you want to solve a problem, you want to find a solution. Okay, you either hunt for solutions that are already existing.
Right.
Yeah. So rank them, which ones are the best ones, and then you
Apply.
have your solution here. Okay.
Right.
So in that sense, that second part is an engineering problem. Then the next part of it is the commerce, basically. Like once that you have done this one, how do you monetize it? How do you actually go through all the regulatory frameworks and get the
drug into the market.
Drug into the market. So we are solving the engineering problem. We are not into the science problem, neither into the next regulatory problem as of today. So we partner with potential
Pharma companies.
partner companies which can do the clinical development.
Understood. So that's still downstream. So now
That's
you
still
mentioned
downstream.
about the science earlier also,
The-
right?
the-
So where do
the-
you partner with the science
Okay.
folks?
For the science, that is what the in-house team we have, in terms of we have Sridhar and we have one more head heading the biology group, Srinivas, and so on, who really look at an indication from ground up, saying that, okay, for example, one of the first drugs that we were going behind was in a condition called Duchenne muscular dystrophy.
Yeah, DMD.
For
right?
DMD. So for DMD, the first part of the journey for us is like where the biologist analyzes the problem and say that, okay, these are very different, varied ways in which we can solve this problem, or we can address the side effects from this problem. And then comes up with a couple of targets, sometimes 1, sometimes 4 different targets, some 4 different pathways that we can go behind. And then is when the engineering takes over. So you start with an indication, you figure out what all can be done on it.
Yeah.
And then actually find solutions, basically find molecules that can modulate these pathways. Validate it using CROs, using basically the larger ecosystem that is where get chemistry synthesized from people, buy from very large virtual libraries, which comes in like there are companies like NMN, Molport and so on, which give us these molecules. Test it in relevant assay systems. And then once you have a data generated on it, that is all done by Peptris, then you license it basically on the value of the data that you have generated. Yeah.
Fair enough.
So our idea is not just to do predictions, saying that how we can modulate a particular protein or a particular pathway with a molecule that AI has predicted. No, but to go forward, actually do the validation effort, ensure that it is actually doing what it is supposed to be doing, first at a biochemical level, next at a cellular level, and at a relevant animal model level. For example, if you are talking about elevation of a disease, actually take a diseased animal, treat it, figure out whether it is doing any good. And that is when we have a data package which is compelling for somebody who thinks that, okay, this can be developed forward into a drug.
Fair enough.
Right. So from our perspective, this entire journey, I think, where is the AI benefit, right? If one were to do the traditional way, like I said, it will take us at least 4 to 5 years to get from identifying that target to work on to a molecule which reaches the humans. With AI, we are able to crunch this time down significantly, maybe to 18 to 24 months. We are able to come out with chemistry which is very different in terms of novelty, right? So you have AI coming out with chemical structures which one would normally not have associated with a solution for that particular protein.
So completely out of the box.
Completely out of the box, because it is mining a space which is 10 to the power 60.
Right.
We have now, for us, we are using a subset of about 1 billion molecules, which we use our compute on. But even 1 billion molecules is a
massive number.
Very large number.
Massive number.
So the diversity that you have in terms of chemistry is significant.
Right.
So the value add that AI brings is not only in terms of crunching the time, but it also brings in that diversity factor, which allows us to look at intellectual property space, which is very different from what was traditionally done.
Fair enough. So, is it safe to say that this fabulous model is kind of fairly new? Is it like, do you think there are comparables in the world that exist with other companies, other startups?
So technically the fabulous model is
in this AI drug discovery.
In this space is pretty new, particularly because most of these systems to support a fabulous model is getting built now. Yeah. For that matter, if say in 2017, when we first looked at say the number of potential purchasable molecules, they were just about 1 million. Then they slowly climbed up, and in the last say about 6 years or 5 years, I would say, right from say 2020, 21 onwards, the number of molecules are kind of like reaching figures which are like more meaningful for drug discovery. Because earlier people used to like look at very niche small libraries of say a few thousand compounds, then it went to 1 million. Only the large pharma had access to them, and so on. Now it has also been democratized. So these are now molecules that are available to much smaller companies, and so in that sense, so we exist not just because we were, so the same concept, we couldn't have done it in 2001.
Right, that's what my
neither
question
could
was.
have done it in 2015 for that matter. And probably the ecosystem is also growing with us. The ecosystem is also gearing up to solving the problem in very different ways. And the AI piece is one part of it, of course. But we also have to acknowledge that the chemistry synthesis space, the robotic, the biology part of it, the amount of improvement that are happening there, the kind of readouts that we're able to get in. Like earlier, what could have taken somebody like in the 2000s or 2010 onwards to get say 5 or 6 different signals out of an assay, we can probably get 100 signals today, as in like biomarker signals also by using the latest of the technologies, so which are not even available 10 years back.
Correct.
Right.
So all of it all of it is changing around. So we are living in very interesting times. Yeah. So this acceleration that we are doing, I would say that we are dependent pretty much on the ecosystem to do it. But asking the other question back, saying that are we one of the very few companies doing it or so on? Across the globe, I would say that there are a lot of companies doing it. Probably out of India, trying to do pure discovery out of India, using all these things that are available. For example, when I talk about the large libraries of compounds and so on, they're not coming out of India. They're coming out of Eastern Europe.
Right.
Yeah. It's coming out of the US that we are doing. And of course, AI as a tool and the AI this one, it is like, it's a global property.
Right. But at some point in time, do you envision that India will also supply to these, with like this database, right? This is the molecule database that's coming to you to do additional work.
You need a lot more investment. You need a lot more groundwork to be done if you have to set up labs like that, which will say that they are prepared to make a billion different molecules or a 50 billion different molecule synthesis automated in a particular way. We need that ecosystem to come in. The ecosystem is still not there.
Right.
So when we talk about ecosystem, so what are we really talking about? So most of them like tell the word ecosystem in a very loose way.
Right.
Now when you talk about drug discovery, when you talk about an ecosystem here, for me, I would say that we are dependent on likes of say NMA. So that's just one of the companies which is doing molecule synthesis using robotic labs. Yeah, and they are able to say that they can make 50 billion molecule library, and you can screen them. You can screen them as in like virtually, and then pick and choose, and then they will supply you those molecules. That is an ecosystem for me. It doesn't exist.
Right.
But we also have to think about like building out of India. Neither are all the cloud infrastructure and other things built out of India. We're still using these to build, right? So like that, when you build a biotech company out of India, we need to think about what is the state of art across the globe.
That's right.
What can we leverage from across the globe?
Absolutely. There's no two way about it.
And like we use the cloud servers of the Amazon that are sitting out of the US 10 years back, we are using these libraries which are sitting out of in the Eastern Europe or in the US now. And we will definitely change over. There's something that will get built here, something, but it's not.
But we are just advancing collectively this whole universe together. Got it.
The other is also the affordability, right? For startups like us, the likes of Enamine, you can get a molecule for about $100, let's say, on an average price, right? Which roughly translates to about, maybe 10,000.
So they ship the molecule to you?
They ship the molecule to us, right?
But you still have
this is in milligram quantity.
Right, and you still have to manage it here, right?
That is all fine. I think that aspect is fairly well taken care of. The logistics is not a problem.
Right.
But if I were to employ a chemist and try to make that same molecule, it would take me a few weeks to make one molecule.
Right.
And it would also incur a cost which is, maybe 50,000 to 100,000 rupees.
Right.
So I'm getting the same service. Now I can go to an Enamine and order 100 molecules, and all 100 would come at the same time.
Right.
Whereas if I were to do this in India, it would take me a longer.
So this is like a little global supply chain that exists for this.
Absolutely.
So you can do that, and I think that's a fair point, because I think what you've decided to perfect is this drug discovery aspect of it. So this is an ingredient.
So we are not investing on building those kind of stuff, right? So we are using that ecosystem.
Right.
The other is, mice are not men and men are not mice. So you can test it in animals. But the availability of humanoid systems, and I'm using that word humanoid a little loosely, right? Where I'm saying if I can have an in vitro system which mimics what will happen in the humans.
That's right.
So now there is a significant advancement, even in India, on 3-dimensional models, which are more human representative than what you would see in a mouse. Right? So you are able to now modulate, actually bring out disease states in these 3D models and test them, which are much more human.
And these are biological 3D models?
These are biological 3D models derived from human cells.
Right.
So it gives that much more relevance to your findings.
So this is like, when you can grow an organ in a petri dish.
Correct.
And then you can test on it.
Right.
Without compromising a human being or needing a human cadaver which is not going to respond in the real time.
So you're looking at advancements like this, which are actually contributing to how we could make drug discovery process much more efficient, much more affordable, right? So if you look at traditionally, they are saying, anywhere around $2 to $3 billion is the cost of developing a drug, which is an amortized cost.
Right.
If we can do this in India, we can bring that cost down significantly. Right? Not because we are cheap labor. I'm not saying that. We're talking about using the ecosystem in a much more efficient manner, which allows us to develop drugs faster and in a much more affordable.
Okay, this has been like an awesome masterclass. And now I think it's time to kind of get into the foundational algorithm of Peptris, right? Like that's the real story. So I want to understand that from you, Anand, because I know you're the kind of I think you're the wizard that's putting all of this thing together, right?
I mean, it's been the discussions with all the people that, when they trickle down, you get the ideas to do something.
Sure.
Yeah, it's one of that. So the crux is something like this. As we explained this, right? We have around 10⁻63 possible keys, molecules that we can actually figure out.
Right.
Amazingly, they're all built using some simple 10 organic atoms, right? And they have been interconnected, and you can figure out what a molecule is by looking at how it is actually bound together. But whatever data set that we have in the public domain, they are very sparse in nature. So you use it, build an algorithm, you can predict something, but it cannot be generalizable because the space is so huge and your limited data set is so small, right? So what we really needed was an algorithm that understands molecules in its entirety. I think nowadays it's very easy to give me an analogy because ChatGPT did the same thing with English.
Right.
A lot of English words, nobody had to tell what it is. You just pass through, understand the meaning of a word based on how it occurs in the context.
Right.
In a very similar way, what we did is we took a very large amount of molecules. We know how they're connected. So we built a language to represent them so that I can write it down in a simple linear way. There is also a language traditionally used called SMILES. It tells how an atom is connected to another atom and so on.
Right.
But it comes with its own disadvantages, the inability to transfer that knowledge to a network and so on.
Right.
So we had to build our own syntax with the knowledge or understanding as to how it is being used in a drug discovery workflow. That is an experience, when we started building, talking to Sridhar and others, we at least have some ideas to how it is being used. So what we did is, we take a molecule, we have our own language syntax to represent it. So you give me a structure, I represent it in my language. Now I have a problem that is chemistry converted into a language problem.
Right.
I can deploy all that we have learned with other
So this is the molecule language that you have created to basically represent these molecules.
Right.
For the system.
For the system.
Right.
And now I can use ChatGPT like an algorithm to learn in a very unsupervised way. It means I don't need anybody to tell me what they are. If I build it, what do I get out of it? Is a mathematical representation that tells me what a molecule is about. Right? And this is the very crux of it, because at the end of the day, what you're trying to do is, I want to ask whether a new molecule, is it similar to another molecule? Will it have a property that I'm looking for? Say, will it bind to a certain target? Will it have a drug-like properties? Will it be soluble? Will it enter into our system? And so on. There are tons of properties that a biologist is really required to know about whether he can progress it or not. And this language or this condensed information that is there in our network helps us to do that very effectively. And this has been our foundational model, out of which we discover many other small sets and then build an automated platform. But the intention is to understand the chemistry in a way that a network can really digest and use it, is where our value addition has actually come from.
Got it. So say it again in terms of the chemistry, like what is simple terms?
In a simple terms, we have built an algorithm to understand the chemistry space as it is. Chemistry space is huge,
right?
But an algorithm understands it, and effectively, you can query that algorithm.
Understood.
Just like you query a ChatGPT, you can query that and figure out what else is possible.
Understood. So you at the end of the neural network pass through of this molecule language that you have created, you basically get some kind of quality data on what properties this will have.
Okay. So it's still an like a representation,
right?
Which you can query and find out what property will it have. Will
right.
it be soluble?
Right.
Will it enter the system?
Right.
Will it be blood-brain?
Right.
Will it cross the blood-brain barrier?
Right.
And so on. So tons of this you can
Tons
query.
of this you can do. And then there is this generative part of it also.
Yeah.
What is that?
What you have learned is from that we have synthesized around 50 billion molecules, say from NMN. But the space is even larger. There is no exploration of that space yet. But if I have a language which is in a mathematical construct, just like a ChatGPT can give you a newer English paragraphs, I can ask my network to produce newer sentences that have not been there anywhere. So it's an avenue for me to explore the unexplored part.
So got it. So this is literally like the ChatGPT first 2022 movement and then you're going to move to the next one till the time you get to like a 5.2.
Correct.
So there's going to be an evolutionary path
yeah.
to the Peptris Foundation model.
Correct, of course.
That will keep moving
Of
like
course.
that,
Of
right?
course,
Is that
of
safe to say?
Of course, yeah.
Okay.
So first the representation, then the generation, then there are more nuanced model like human feedback. When it comes into the loop, it becomes a very powerful model for us to actually believe in.
Got it. Now, help me also explain this part that, Venkat touched about. This is like really about the sort and search problem, right? So there is this sort and search, then there is this posing of the 3D model and then predicting what it will do, and then you are optimizing the parameters, right? Like how does that process work, and then what's the feedback loop in that?
Yeah. So I would give an analogy again. Trying to find a good molecule is like trying to find a gold in a very large area. Right? I don't know where the gold exists. You need a scanner to find out where all it exists.
Right.
So the first problem is about search. What we are dealing with is say a 30 billion synthesizable molecule. I need an algorithm to search. You put a computational algorithm, a costly one. I don't think there is any brute force way to search through all of them.
Right.
You need a smarter ways to search, and that is what we call ourselves of the explore phase, where these algorithms go search 30 billion and give me a possible list of.
So how many algorithms like this you've made?
We have 3 or 4 in this bucket,
right,
which is we call as explore,
so
predominantly for a search algorithm.
Got it.
Foundational model is a very good one because it gives me a fingerprint. I can go and search who is the most closest one.
Right, that's a match.
That is a match. That is one of it.
Right.
It is not a very accurate one. I need a most probable answers in
that's
30
right.
billion.
Right.
The intention of the algorithm is compute fast, give me a list of molecules. You don't need to be extremely accurate.
Fair enough.
Right. So that is the explore phase.
Right.
Once I have a possible list, I still need to make it a very good accurate answer. That's where the sort algorithm comes. I take these 1,00,000 molecules. I want to sort them so that they are much accurately so that I can pick the top ones.
Right.
In this phase, we can put more computational power. I can take a 3D shape of a protein. We can take a 3D shape of a molecule, fit them in, figure out as to how they are going to interact, and then score them.
Got it.
So this is what we call as an enrich
phase.
Where you able to enrich the initial set into a much finer,
more
set.
complex data points, more complex structures.
Right. But at the end of the day, from 1,00,000 now I'm coming down to thousands.
Right. So it's a filtering mechanism.
As a pipeline goes through.
Right.
The third stage is also very interesting because traditionally, once you have some kind of hit, chemist sits on that molecule, modifies it because they know that what changes are good for body. Will it become a drug-like molecule?
Right.
Will it be soluble?
Right.
It is not just about trying to hit a target.
Right.
It's also about whether it becomes a drug. Right. So medicinal chemists have this knowledge internally and then they modify it.
Right.
Now we use our Gen AI algorithms to modify the molecules and fit those properties.
Right.
So it's not a single parameter, it's a multi-parameter optimization that we employ, which we call as an evolve phase, where we start modifying and then we get molecules which are highly enriched and then possibly have a good answer or looking for.
That's awesome.
So if you stitch all these three, we get automated pipeline.
Got it. Okay, so that model explains really well, but the idea is that not just the language and the foundation model, but also the algorithm, which is also the orchestration platform for Peptris, they kind of evolve simultaneously. So that's the vision, how you're kind of building and scaling the whole organization. Now, very interestingly, obviously, you have run through the proof of the pudding is out there. We have Pepper 124 that you have actually licensed. So congratulations on that. Would love to know that journey of identifying Pepper 124 for DMD.
So one of the aspects that we do in addition to finding novel molecules is finding new uses for existing approved drugs, right? So the algorithm is built in a way where it identifies off targets for a particular molecule, which means these are targets which is not the intended primary targets for the molecule. So you have a primary mechanism, and because the proteins are so diverse, or there are certain families which may be close to each other, you may hit multiple of those, right? You may fit some of those, but may not be to the same extent or potency that you fit the first intended target.
Yeah.
Now, when you look at this vast range, some of those could potentially have uses in diseases which have never been looked at. So that is how Pepar 124 came into being. So this molecule is one which is currently in the market, approved for maybe the last 2 decades for a diabetes, right? And using our AI algorithm, we identified targets which could potentially be impacted by this molecule, which could have a role for Duchenne muscular dystrophy. So these are 2 very distinct disease buckets.
And nobody would have thought of...
Nobody would have a priori thought of this at all.
Your diabetes would connect to muscle.
So the utility of AI came in there in identifying what are those potentially novel targets that could be, implicated using AI, and how that particular molecule could then be tested for a disease like DMD. So AI throughout these targets, we looked at what those targets meant for a disease like DMD.
Right.
And we went through the testing, the in vitro testing initially, where we saw significant changes with this molecule. We then did a 28-day mouse model for Duchenne muscular dystrophy, and we were very surprised with the kind of effect that this molecule had, beneficial effect that this molecule had. And now we licensed this to a company called Revio Therapeutics, and they have now progressed this molecule to a 90-day study where we have reproduced what we saw in the 28-day study, showing that chronically it still works. They're now moving it into clinical development. We are filing, since it's a repurposed drug, there's a significant amount of safety information that's available. There's significant amount of human data that's available, which shows that this molecule can be safely administered to humans.
So this will actually accelerate the path of
It is going to accelerate the path towards clinical development and commercialization.
Right.
So they have now filed for a IND request with the US FDA. We have got an orphan drug designation from the US FDA on this one, which would ensure that this gets fast-tracked. And through the regulatory pathways, and can actually become a drug within the next 5 years.
Wow.
For treatment of DMD, which is a very high unmet medical need.
Yeah, and Peptris would have had the significant role to play in this.
In identifying this molecule and its utility for DMD.
Amazing. No, this is just the beginning, and we're just scratching the surface, I think.
Absolutely.
Because there is so much of possibility.
Yes.
So this is a whole universe that remains to be explored. Great. So, and a wrap up part of it, now that we have had like this double down conversation on Peptris, love your reflections on the ecosystem that we have here, right? We're sitting right in the middle of the Bangalore Life Sciences cluster.
So it's an amazing place to be. Good, happy that we discovered this. Yeah, and if you look at it, when people talk about ecosystem, the other time I was talking about like what is a real ecosystem. So when you come to a place like C Camp here, yeah, so it is not about just providing the lab spaces or providing a place to do work and so on, but actually having a good set of startups in the same place actually talks about like kind of cross-pollination of ideas, trying to figure out what we can do. They also try to get in the investors in here. So we also get mentored internally as well as pitch to investors across who are coming here. They also get in the regulators once in a while, saying that, okay, there are key opinion
developments
leaders,
and
key opinion leaders from different parts of it, patenting or regulatory part of it, they coming and giving us talks and so on. So
the IP laws need
the IP
rejig.
Laws. So all these things actually make this place a very vibrant place.
Right.
It is not that you are here just for the
infrastructure,
the infrastructure or the beauty of this place, but also all these others, the salt and pepper that gets added to it, extra to it,
and the academic connection. InSTEM is right here. Everything is right here. So you can do so much of multidisciplinary magic in just this
and that makes
few
a
acres.
huge difference. So when you talk about ecosystem, you really talk about does it have all the ingredients to make it a place where you can grow and thrive.
Right. No, this is awesome. I love hanging around here also because of just the beautiful landscape that this campus is. This has been an absolute masterclass on AI in biology. I think I'm not a Tetris fan, but I think I'm definitely a Peptris fan. So really wish you all the best for next maybe 1 million molecules to come out very quickly from the foundation models here. So thank you so much today for joining me for this Double Down Pod episode. Thanks, Venkat.
Thank you.
Thanks, Anand, and thanks, Sridhar.
Thank you for the conversation.
Yeah, thank you so much. Thank you so much.
Bye-bye.
Bye.